Happy day everyone. Today in this video we are going to learn about biomolecules. Let's start. Now, what is biomolecule? Firstly, we have to understand the term. Bio, whenever you see the word bio, it means living system. Okay. Bio means living. So, biomolecules are nothing but the molecules which are containing carbon in the living system. So, in short, carbon containing compounds in the living system. Now, we are clear what does it mean by biomolecules, and all these carbon containing compounds are formed with the covalent bond. Now, we understood the term what is biomolecules. Now we talk about another thing. So let's say if we take earth crust and living tissue and compare what are the organic compounds present in that, we could see both are having carbon, hydrogen, nitrogen and silicon. Then what is the difference between the living tissue and non-living tissue? If both are containing the same, the difference is the amount, the quantity it contains. So in case of living tissue, carbon and hydrogen will be higher in number, but in case of earth crust, the carbon will be not in number like quantity, but the carbon will be of less quantities. So but in case of earth crust, the silicon is of high quantity, but in case of living tissue, the silicon is will be in the negligible quality. So this is the difference. Even though same amount of organic substance are present, the quantity makes the difference between the living tissue and the non-living tissue. Now we will come to another part. Now let's say I am taking a piece of vegetable. What are the organic compounds present in it? Can I say it just by looking? I can't, right? So we need a special method to find it out. What is that method? Let's take a piece of vegetable and we will be adding TAA, which is nothing but trichloroacetic acid, and we will grind it using the mortar and pestle. Okay. While we are grinding it, we will get a thick slurry. Once we obtain this thick slurry, we will be filtering it out using the cotton. Let's say whenever you are filtering out any juice or something which is homemade, you will find only juice will go in the below and above stuffs like seeds will stay in the above. The same will be happening here. Once you filter the slurry with the cotton, there will be two types of fractions, we could say. One is macro and another is micro. This micro is called as filtrate and the macro is called as retinate. So what is filtrate? The particles which are filtered out like juice. What is retinate? The particles which return like the seeds of the juice. Okay, now we are getting it. So these particles which are coming in this filtrate are called as micromolecules, and these particles which are remaining in the retinate are also called as macroparticles, and it is also called as acid soluble or filtrate, and the macroparticles fraction is called as acid insoluble or retinate. Now we are clear how we are doing the chemical composition, like how to find it. We will be adding trichloroacid and we will grind it using the mortar and pestle. Once we get the slurry, we will be filtering out and we will get the two fractions. One is macromolecules and one is micromolecules. So there are two types of biomolecules. One is microbiomolecule and macrobiomolecule. I hope you are clear. Now we will talk about the another part. So this human or a living organism only contains organic compounds. There is no amount of inorganic compounds we do have. So this testing is especially for the organic compounds, right? So how we will know what are the inorganic compounds present in us? For that, we will take a leaf. Okay, this leaf is called as a wet weight. Once we dry it, drying in the sense once all the H2O is absorbed, then it is called as the dry weight. We are taking a leaf, it is containing the wet weight. Once we dried it, H2O will be gone, then it will become a dry weight and we will burn it. Once we burn all the carbon containing compounds, so what are the carbon containing compounds? Nothing but the biomolecules or organic compounds will be removed out of the material, and the remaining is called as the ash. Okay, and it contains the inorganic compounds. You are getting, once we get the dry weight and we will burn it. Once we burn, all the organic compounds are removed from it. So what will be left? Only inorganic compound, that is called as ash. Together it is called as ash, and this ash contains calcium and magnesium, etc. Few inorganic compounds such as phosphate and sulfate are also seen in acid soluble fraction. Which is the acid soluble fraction? This filtrate part. These two are also seen in the acid soluble fraction. Now we saw about the micro and macro, right? Acid soluble and acid soluble. insoluble. So it is nothing but if you say micromolecules, take amino acid. This is the monomer, right? the monosaccharide. And how is this macromolecule formed? Nothing but the addition of amino acid. The addition of micromolecules will form the macromolecules. Agree? Say, if here the micromolecules is amino acid and macromolecule is protein. How the protein is formed? By the addition, like a chain of amino acid. So macromolecules are nothing but it also formed by the addition of micromolecules, and this micro will come under the acid soluble and the macro will come under acid insoluble. So I hope you are clear with the introduction. What is biomolecule and what is the difference between the non-living and living tissue, how we will test the chemical composition, and what are the inorganic compounds.